Yeah, I have no disclosures, by the way. I am in Rotterdam already long, in Utrecht, and also in Cologne. But I would like to talk about how DNA damage leads to transcription stress and then subsequently to proteinopathies, as in dementias, and the amazing impact of nutrition. And I hope that you'll share with me that the story is indeed really unexpected. So I work for a long time already on DNA damage. Damage inflicted to DNA can come from outside, UV, radiation, chemicals. It can also come from our own metabolism, and the ROS, but numerous other factors, water, mechanical stress, all damage DNA. And of course, nutrition is very important because nutrition comes from outside and enters our body and is used. Of course, we expect that every cell in our body every day may experience up to 100,000 or more lesions per DNA, per day. So, of course, DNA metabolism will be altered, and there are two very different but important consequences. One of them is that damaged DNA is nevertheless used for replication. That leads to mistakes, mutations, chromosomal aberrations, and that causes cancer. So DNA damage is the main cause of cancer. But other types of damages that block transcription can give transcription stress, or that arrest replication can give replication stress. And that leads to cell cycle delay, maybe a permanent cell cycle arrest. That leads to senescence and also senescence-associated secretory phenotype, including inflammatory markers. Transcription can go down, function be affected, and cells may die. All of these, we think, are the main factor for systemic aging. And fortunately, there are repair systems that are constantly trying to repair the damage before it leads to either mutations and cancer or to cell death and senescence and aging. And it is important to realize, even I have no time to explain the repair systems to you because it would take too much time, but even with a normal, efficient repair, some damages are not detected by repair, and some damages are irreparable. DNA is too damaged. Sometimes repair is too late or makes mistakes, and repair may itself decline with aging. So still, DNA lesions inevitably accumulate in time, causing cancer, and we think also majorly contribute to systemic aging. And of course, when you are born with the repair defect, then all of these events take faster. It goes faster. So I would like to show you one of those diseases. There are many of them. They are called defects in a pathway, which I also have no time to explain. It's called transcription-coupled repair. When transcription is blocked by damage, that this repair system tries to solve the problem. And there are very rare human syndromes that are linked with defects in this pathway. And one of these is called Cockayne syndrome, very rare, 1 in a million. These children have many problems. Peculiarly, they have no cancer or have less cancer than normal children do, but on top of that, they have severe neurodevelopmental abnormalities and a life expectancy of only 12. This is Baptiste, a French patient, 3 years old. You see already neurologically not very much abnormal. 4 years later, he's very small, needs glasses, needs hearing aids, cannot walk, cannot talk. And Baptiste became only 10. And for comparison, this is his sister. He is 1 year older. So you see the dramatic phenotype that these children have when you are born with a defect in this transcription-coupled repair system. And there is no medication, but I'll come back to that later. Another disease, many elements in common, but still very different, is called trichothyroidystrophy, also very rare, 1 in a million patients. So but they have many features in common, developmentally and so on with Cockayne, but on top of that they have brittle hair, brittle nails, scaly skin. And these syndromes, they are due to, they have a multi-organ accelerated aging phenotype, primarily focused in the post-mitotic organs, let's say the neurons, the liver, the kidney, the skeleton, but for instance the hemopoietic system is more or less normal. So this is what we call segmental aging, and in order to try to find a solution for these diseases, we generated many mouse mutants for those diseases. And all of these diseases are genetically heterogeneous, so we have made many different mouse mutants and other repair mutants. We are the ones in the world that have generated most of them. But these diseases are specifically linked with something with transcription. I'll come back to that later. So we made mice. For instance, this is the TTD patient, and this is the mouse. The mouse has the same point mutation in the XPD protein, that's a repair protein involved in transcription-coupled repair and also other repair systems, like the patient. And indeed, as you see, the mouse has also brittle hair, brittle nails, scaly skin. And the mouse actually showed us that these children, without us realizing at that moment, were aging. Because you can see kyphosis, you can see a frail phenotype, cachexia in the neurological degeneration. So actually the mouse revealed to us that these very rare children actually were aging faster. I have no time to show you all of the evidence for this, but the aging is, again, segmental. It is only particularly in the post-mitotic tissues of the body. So we wondered, so we have this mouse that can become still 1.5 years. All of the patients with that mutation died before the age of 5, so it's a very severe disease in humans. So we wondered, the mice can become 1.5. What would happen if we make the repair defect more severe by crossing the mouse with other repair mice that we have made? So this, we crossed it with an XPA mouse. That's a cancer-prone mouse. The XPA mouse shows not so much aging, but mostly cancer because of a repair defect. The TTD mouse dies of accelerated aging, actually overprotects from cancer at the expense of too much aging. So they have less cancer, but more aging. They become 1.5, and the other mouse, the mouse to the left, that's the double mutant, and that mouse lives only 5 to 8 weeks. It gets already kyphosis at a very early moment in life. So the severity of the repair defect determines the acceleration of a segmental aging phenotype. So this is a double mutant. And this is not the only one. We have made many mice, cocaine A, cocaine B, XPA, XPC, XPG, XPB, XPD. So all of those, and they boil down to the very same message. The more severe this repair pathway, the faster the aging phenotype, but segmental. Now I'll show you a mouse where more repair defects are affected. In this case, that's the ERCC1 mouse. We were the first to clone a human repair gene, ERCC1, and this gene happened to be involved in 4 pathways. And now we see aging in other tissues that are very much depending on the pathways that are affected in this. For instance, the cross-link repair gives a bone marrow aging phenotype. And here is the mouse. I think the champion in terms of accelerated aging, in terms of widespread multimorbidity. This mouse shows aging in the neuronal system, dramatic, I'll show you later, but also kyphosis, osteoporosis, vascular stiffness, hypertension, cardiomyopathy, kyphosis, the liver, the kidney, the skeleton, the fat tissue, the muscles, the skin, urinary incontinence, stem cell exhaustion in the hemostatic system. So this is the typical multimorbidity, because now we have affected other repair systems that are critical for other tissues to be prevented from accumulating damage and aging. So this mouse lives 4 to 6 months, and it shows the typical multimorbidity that you also see with natural aging. So you never age only in one organ, you age overall. And this mouse, and it shows what spontaneous DNA damage, if you cannot properly repair it, can do. And actually, even when you can repair, then still damage persists. So this mouse is a role model for neurodegeneration. These mice lose vision, they lose hearing, they get reduced synaptic plasticity, cognitive decline, tremors, ataxia, imbalance, neuronal loss. They show dramatic overall, this is the brain of an 8-week-old ERCC1 mouse compared to a wild type, stained for GFAP, activated astrocytes when neurons are not doing well. You see brown staining over the whole brain of the ESCC1 mouse. So everywhere neurons are signaling, I'm not doing well. And actually, if you look at the graphs, at 4 weeks, there are already a few animals that show P53 and are dying, and at 16 weeks, even more. So it's progressive. And these mice show brain shrinkage, ventricular dilation, loss of neuronal plasticity, neurons, motor performance, memory, hearing, vision, wherever you look. And when we take full genome expression profiles of the brains of these mice and compare that to human profiles, one of the top genes, top diseases that pops up is Alzheimer and Parkinson. So, and also the development of the profile in time follows the development of Braak stages in Alzheimer. And also, if you look at the critical genes, they are all the same way dysregulated in ESCC1 brains compared to Alzheimer. And also metabolites show the same trend. So this captures a tremendous amount of underlying biology between ESCC1 repair mice and Alzheimer mice, and Alzheimer in humans. So we think that is entirely consistent with the notion that the main risk factor for Alzheimer is aging. Parkinson too. So I'll come back to that later. I'll show you we can also, so we think this mouse is a valuable model for Alzheimer, addressing a tremendous medical need. We can also target the repair defect only to one compartment in the brain. So now you see brown staining in the forebrain and in the hippocampus, because now we made ESCC1 deficient in that part of the brain only, the rest not. The rest of the body is normal, but these mice, traumatic neuronal degeneration in the cortex and the hippocampus, and learning motor performance decline after the age of 17 weeks. So not only, we can also do that in different, we have made other mice, and you can see it also progressively. So on the top line you see a wild type, then an XPG mouse, that's an other repair mutant, one repair defect less compared to ESCC1, and a slower decline of the hippocampus at 26 weeks XPG, and then following to right is ESCC1 in half a year, and in 1 year there is hardly any hippocampus left in the ESCC1 cortex for forebrain-specific knockout. So the dose-response relationship indeed also is consistent with causality. And this is similar to what happens to Alzheimer hippocampus, as you can see in this slide. So not only the hippocampus can be targeted, but we can also target the Purkinje cells in the cerebellum. As you can see, the brown staining now is only there, and if you look closely and zoom into Purkinje cell layer, you know, in time you see more and more Purkinje cells disappearing, and after 1 year there is hardly any Purkinje cell left in this mouse. And so we can follow what happens in the rest of the body when you functionally deteriorate in all of these specific organs. It can also be the heart. I could show you also movies of heartbeat, or the liver, because we can target aging to any specific organ. What we also found, and that was a very important observation, we have made many mice that show accelerated aging in different age rates. I showed you only a few. But if you look at the expression profile in the liver or the kidney or the lungs of these mice that show accelerated aging and compare that to age-matched wild type, then very clearly the short-lived repair mutants suppress the growth hormone, IGF axis. So that axis, the somatotrophic axis, the thyrotrophic axis, the lactotrophic axis, they are all axes are down. And they upregulate resilience mechanisms. The antioxidant defense is upregulated. Stress resistance is improved. And the low IGF levels explain why these children stay very small, because they also suppress their growth. Instead, this profile resembles a profile after dietary restriction. If you have not enough food, then the body also doesn't invest into growth. It invests into maintenance and resilience. We call that a survival response. And dwarf mice that are born with a defect in the axis, they stay small but live long. So they have delayed aging. So paradoxically, a mouse that shows dramatic accelerated aging shows a response that resembles anti-aging, dietary restriction, delayed aging. So So paradoxically, DNA repair deficient, DNA damage-driven accelerated aging, as well as normal aging, trigger an anti-aging survival response upon dietary restriction. So we wondered, these repair mice, they have already a response that resembles dietary restriction, but what would happen when we would actually give them less food? So, and that was an amazing observation. So this is the normal lifespan, in this case females of ESCC1 mice that I showed you before. They are delta mice, they are not complete knockout, but it doesn't matter. So the first mouse dies at the age of 12, if you look closely, and the last one dies at the age of 27. And now the other half, so they live about 4 to 6 months. The other half we put on a diet. So from week 7, 10%, 20% in week 8, and 30%, and then on a restriction. And then looked what happened. And then the mice lived 3 times as long from the moment that you put them on a diet. So 200% lifespan extension, median as well as maximum lifespan for mammals, unprecedented. So these mice respond very well to calorie restriction, which is an anti-aging intervention. So, and not only they live longer, the liver, the kidney, the immune system, the retina, the neuronal system, all do very much better. So particularly the neuronal system benefits. That benefits most. Dramatic improvements. I'll show you 2 movies to document that. So these are 2 littermates. The one on the right had food all the time, is neurologically already so much compromised that it cannot normally walk. Neurologically, the muscles are not controlled anymore by the neurons, and you can see the mouse can have these spastic contractions, these tremors. It cannot keep balance. So neurologically, So if you put these 2 mice, the one that could run on the other hand and on a rotating rod, well, the difference couldn't be bigger. The one on the diet can run. The one that as ad libitum food falls down. 3 minutes later, a fat wild-type mouse drops, and now the wild-type on diet drops. So the difference between the 2 is just food. 30% less food, and the first one drops in the first round, and the one on diet can even stay as long as the wild-type. So if this would apply to children, they might become 36 and would be in a much better shape. But of course, mice and humans are not the same. So, and these children are fragile, and they get extra food. So we argued these children should get less. Well, it took 3 years to convince clinicians, maybe we should give it a try. And now I show you the first patient. This is Emma, a child that has TTD, and I don't hear the sound. Yeah, so you see brittle hair, but she barely can crawl. She shakes like the mice. 6 years old, gets every day 1150 kilocalories directly in the stomach because she has no appetite. Now, Emma developed tremors. The whole body starts shaking, as we see in the mice. She could not put 2 duplo blocks on top of each other, a very bad prognosis, because then you get paresis, and then they die. These children, many of them die before the age of 10. So, unfavorable diagnosis, we decided to gradually lower the intake in steps of 100 and see what happens. You can always go back. We started 28 May 2018. Within 12 days, Emma could build with Duplo again. The tremors were gone. Then, a few months later, she started to be able for the first time in her life, at the age of 8, to walk independently. So then I got this movie. She, for the first time, could pronounce words. Before, there were only sounds, no articulation. Then she started to write her name, Emma. She didn't need diapers. She could build these constructions. Now she's on a bicycle. And she survived COVID and enjoys life. So for more than 4 years now, she is, and I expect her, I would not have expected her to stay alive. Well, of course, this is all very important. It's better than any medication would be expected to accomplish in this genetic disease. But what is the mechanism? Well, unfortunately, I have no time to show you, because we think we understand the mechanism. And we think that it is also extremely relevant for Alzheimer's disease, Parkinson's disease, and other dementias. And nutrition keeps a tremendous promise for treating patients in an early stage of their dementia process. So I have to skip a lot of slides because I'm overly optimistic. And show you about DNA damage causing transcription stress and neuronal dysfunction, and also that transcription stress leads to protein aggregation, and protein aggregation leads to early cell death in neurons, and that explains the aging contribution to Alzheimer, to Parkinson, and to all proteinopathies, because we understand why all these proteins, be it beta amyloid or hyperphosphorylated tau of alpha-synuclein, why all of these proteins start to accumulate with aging because of DNA damage that leads to this derailed transcription, derailed protein synthesis, and in the end, protein stress and protein aggregation. So I'll stop with all of these. I'm really optimistic. And show you the last slide. We find also, by the way, amyloid-like aggregates in our mice, even though they have no mutation in beta amyloid. And that calorie restriction and at least food components might be very beneficial for dementias like Alzheimer, Parkinson, and other proteinopathies. So nutritional intervention and mimetics, perhaps, will change, we think, medicine. Not only dementias, but surgery, organ transplantation, chemotherapy, radiotherapy, all can benefit from less nutrition. So this is our laboratory, not a cook, a look in our laboratory. The mice, the names of many people that I have no time to mention, but I have the privilege to present their work, funding agencies, but particularly this child. Thank you very much for your attention. Sorry for an overtime.
Thank you very much for this.